In this video, we're going to learn about the last stage of aerobic cellular respiration, and that is the electron transport chain. The electron transport chain occurs on the inner membrane of the mitochondrion, and it involves moving ions across the membrane from the matrix into the intermembrane space, and then moving them back into the matrix. And the molecules that go into this process are NADH or FADH2, oxygen, and then ADP and inorganic phosphate. The outputs are NAD plus and FAD, water, and a lot of ATP. The main idea of this reaction is that NADH and FADH2 that were produced in the previous reactions are used to make ATP. And the way they do this is by releasing their hydrogen ions and electrons and passing them along this electron transport chain. So as I mentioned before, the setting for this process is in the inner membrane. And here we have a portion of the inner membrane of the mitochondria. And you can see that there are several carrier proteins that are embedded in the membrane. So these ones don't move. The first one is called NADH dehydrogenase. The second one is called cytochrome BC. And the third one is called cytochrome oxidase. And then there are two mobile carrier proteins. They act like shuttles that can carry things back and forth between the embedded ones. So one of them is ubiquinone, and the other one is cytochrome C. And then finally, the most important and the most interesting carrier protein is ATP synthase, which we'll talk about a little bit later. But this one's really important in producing the ATP. So you'll see here that the lower half of this membrane is where we find the matrix of the mitochondrion. So this would be one of the folds in the cristae. And then the other part up here is the intermembrane space. In the first step of this process, NADH or FADH2 that were produced in glycolysis, the link reaction of the Krebs cycle, bring their electrons and their associated hydrogens to the electron transport chain. And what they do is they reduce the first carrier protein by donating their electrons and by giving up their hydrogen ions. And when a carrier is reduced, some of the energy that is gained is used to pump hydrogen ions through the first carrier protein and into the intermembrane space. This first electron carrier here is known as NADH dehydrogenase, so the name explains what it does. So it takes off a hydrogen and the electrons from NADH, so NADH dehydrogenase. The electrons that were released from NADH are then passed to a mobile carrier protein, which transports it to the next embedded carrier protein. And as before, some of the energy gained by that next carrier protein as a result of being reduced is used to pump hydrogen ions from the matrix into the intermembrane space. Eventually, a concentration gradient of hydrogen ions is established in the intermembrane space. The very last carrier must get rid of the electrons that have been donated to it. So they pass the electrons to oxygen, the final electron acceptor, and in this process it forms water by taking some of the electrons into hydrogen ions. We need 2 electrons and 2 hydrogen ions to form 1 molecule of water. And this process happens twice for every water molecule. Now we come to ATP synthase, where we produce the ATP. And ATP synthase produces ATP by phosphorylating ADP, so adding an inorganic phosphate to ADP. The energy for this reaction comes from hydrogen ions that start forcing their way through ATP synthase, going along with the concentration gradient, and passing from the intermembrane space into the matrix. And this gives the ATP synthase rotational energy, and that rotational energy is able to combine the ADP and inorganic phosphate. This process is sometimes called chemiosmosis. So hydrogen ions moving across the membrane following the concentration gradient will release the energy the ATP synthase needs to make ATP from ADP and inorganic phosphate. And that's what chemiosmosis means. The electron transport chain and ATP production is also sometimes known as oxidative phosphorylation. And this is because ADP gains a phosphate group to become ATP. So that's where the phosphorylation part comes from. And this is done using the energy that's released by the oxidation of NADH at the first electron carrier. The structure of the mitochondria allows for many of its functions. First of all, cristae form a large surface area. So this allows for multiple electron transport chains all along the inner membrane of the mitochondrion. There's a very small space between the inner and outer membranes, so this allows for an accumulation of protons, also known as hydrogen ions, to help create a concentration gradient, so that those ions or protons can come back through the inner membrane by way of ATP synthase. And then the fluid matrix contains all the enzymes for both the link reaction and the Krebs cycle. So all the substances that are required for all of these processes are found floating around in the matrix. So right now we're going to take a look at some of the ATP yields per glucose molecule. So if we look at each of the stage or pathway, we'll look at the direct amount of ATP made and then look at the number of NADH and FADH2 and then figure out how many ATP those would equate to once they get to the electron transport chain. So first of all, glycolysis, we make, if you remember, 2 net ATP. And if glycolysis is where you end and you go into lactic acid fermentation or ethanol fermentation, which we'll talk about later, that's all the ATP you get per molecule of glucose. So not a really efficient process. But we need those processes if there's no oxygen present. But if you remember, glycolysis also makes 2 NADH. And when those molecules end up at the electron transport chain, each NADH has the power of making or allowing 3 ATP to be made. So out of the 2 NADH, that would be 6 ATP. So from glycolysis, the total ATP that can be produced through aerobic respiration would be 8. The link reaction doesn't make any ATP, but it does make 2 NADH, and then in some side process, they get converted to 2 FADH2. So FADH2 are able to produce 2 ATP from each molecule. So that would be 4 ATP for the 2, 1 per link reaction. Remember, glucose breaks into 2 pyruvate at the end of glycolysis, and then we have to trace each of those per reaction. So the Krebs cycle, because there's 2 turns, 1 per pyruvate, you get 2 ATP, 6 NADH, and 2 FADH2. Those 6 NADH can make 3 ATP each, so that would be 18 ATP, and then 2 FADH2 can make 4 ATP. So that's a total of 24 ATP from the NADHs and FADH2s and the ATPs made in the Krebs cycle. So all together, if you add all of those up through the pathways before the electron transport chain, and you get 4 direct ATP made, and then out of all the NADH and FADH2 that are made, you end up with 32 ATP, so a total of 36. Now some places will say there's 38 because they don't talk about this process, the conversion from NADH to FADH2 after the link reaction. So it's a lot of ATP. So it's really advantageous to have oxygen around so that you can produce lots and lots of ATP.